Hello, welcome everybody. My name is Jovana Aleksic and I'm a PhD student at Polytechnic University of Madrid. Today I'll present you our work about prediction of protein-protein interactions, which we did in collaboration with Weill Cornell Medicine in Qatar. So this is our poster for the conference. This part of the poster here is briefly explaining motivation, problem description, and some of the solutions which we proposed. This part of the poster here is explaining the dataset we used for training our model. It is important to note that this dataset was customly developed at Weill Cornell Medicine Qatar by their genomics team. They developed a new method for detection of protein-protein interactions, which is very briefly working as follows. So they take full-length proteins, they shear them into random fragments, then they randomly pair those fragments. They put them in 3 different conditions. One of the conditions is neutral. The other 2 conditions are selective, mild and strong pressure. Then the expectation is that interacting fragment pairs will grow comparing to the others non-interacting. Then all of this is put under pair and sequencing in batches of millions. So finally, we have for each of our data point, we have a row in the table which looks like this. Here we have fragment pair. Here we have 3 replicates for base conditions, 3 replicates for mild pressure, and 3 replicates for strong pressure. This allows us to consider this as a regression problem and not as a classification. According to our knowledge, this is the first work that somebody treats protein-protein interactions as a regression instead of classification. The target value, which we try to predict here, is strength of interaction. Another good thing, which this dataset allows us, is mentioned here, and that is that we can potentially predict interacting region, because we work with fragments and not with full-length proteins. This part of the poster here explains how we model strength of interaction. We do not model it as a point estimate, but rather as Gaussian distribution, and parameters of the distributions are given here. This part of the poster here at the bottom explains main parts of our model, which are convolutional layer and LSTM layer. We use those 2 layers because we believe convolutional layer is good at exploiting spatial dependencies, and LSTM layer is good at exploiting sequential dependencies. Proteins are chains and therefore sequential nature of amino acids, but they also fold in a specific 3D shape, which we believe can be captured better by convolutional layer. We use Bayesian neural networks because we also want to predict distributions and not point estimates, and Bayesian neural networks have built-in uncertainty estimations. On the right side of our poster, you can find a section about data imbalance. It is a problem frequently mentioned in classification, but not so much in regression. As you can see on this graph here, this is a distribution of strength of interaction for our data points. Majority of data points is centered around 0, or majority of our fragment pairs actually do not interact. We believe this is a problem worth of mentioning, and we propose a solution which is oversampling adapted for regression. Finally, this part of the poster here tells us about the biggest problems we encountered and about the future directions for research. One of them is definitely data imbalance, but there are also other problems mentioned. For example, a problem where we didn't have any reads in baseline conditions, but we do have some numbers in selective conditions, and therefore we effectively don't know what is the strength of interactions. And there are also a couple of other things mentioned. Thank you very much for your attention.